scientists have published the first complete gapless sequence of a human genome 2 decades after the Human Genome Project produced the first draft human genome sequence. According to researchers, having a complete gap-free sequence of the roughly 3 billion bases or letters in our DNA is critical for understanding the full spectrum of human genomic variation and for understanding the genetic contributions to certain diseases. The work was done by the Telomere to Telomere T2T consortium. Analysis of the complete genome sequence will significantly add to our knowledge of chromosomes, including more accurate maps for 5 chromosome arms, which opens new lines of research. This helps answer basic biology questions about how chromosomes properly segregate and divide. The T2T consortium used the now complete genome sequence as a reference to discover more than 2 million additional variants in the human genome. These studies provide more accurate information about the genomic variants within 622 medically relevant genes. The now complete human genome sequence will be particularly valuable for studies that aim to establish comprehensive views of human genomic variation or how people's DNA differs. Such insights are vital for understanding the genetic contributions to certain diseases and for using genome sequence as a routine part of clinical care in the future. Many research groups have already started using a pre-release version of the complete human genome sequence for their research. The full sequencing builds upon the work of the Human Genome Project, which mapped about 92% of the genome, and research undertaken since then. Thousands of researchers have developed better laboratory tools, computational methods, and strategic approaches to decipher the complex sequence. 6 papers encompassing the completed sequence appear in Science, along with companion papers in several other journals. That last 8% includes numerous genes and repetitive DNA and is comparable in size to an entire chromosome. Researchers generated the complete genome sequence using a special cell line that has 2 identical copies of each chromosome, unlike most human cells which carry 2 slightly different copies. The researchers noted that most of the newly added DNA sequences were near the repetitive telomeres, that is, long trailing ends of each chromosome, and centromeres, that is, dense middle sections of each chromosome. The cost of sequencing a human genome using short-read technologies, which provide several hundred bases of DNA sequence at a time, is only a few $100, having fallen significantly since the end of the Human Genome Project. However, using these short-read methods alone still leaves some gaps in assembled genome sequences. The massive drop in DNA sequencing cost comes hand in hand with increased investments in new DNA sequencing technologies to generate longer DNA sequence reads without compromising the accuracy. Over the past decade, 2 new DNA sequencing technologies emerged that produced much longer sequence reads. The Oxford nanopore DNA sequencing method can read up to 1 million DNA letters in a single read with modest accuracy, while the PacBio HiFi DNA sequencing method can read about 20,000 letters with nearly perfect accuracy. Researchers in the T2T consortium used both DNA sequencing methods to generate the complete human genome sequence. The researcher says that in the future, when someone has their genome sequenced, they will be able to identify all of the variants in the DNA and use that information to better guide their health care. Truly finishing the human genome sequence was like putting on a new pair of glasses. Now that we can clearly see everything, we are one step closer to understanding what it all means.